Lee Blaney is a professor of chemical and biochemical and environmental engineering at the University of Maryland, Baltimore County. Lee's presentation is entitled Contaminants of Emerging Concern in the Environment, Unintended Consequences, New Chemicals of Concern, and Understudied Sources. The brief abstract is that thousands of contaminants of emerging concern enter the environment every day. This presentation highlights prominent and unintended consequences of these contaminants on public and ecological health. It identifies recently reported chemicals of concern, and it emphasizes the importance of expanding sampling locations to evaluate alternative sources. Dr. Blaney, the stage is yours.
All right, thanks so much for the introduction, Dan, and to all the organizers for putting this together. I think it's going to be a fun event here. And as Dan said, really today I'm going to be talking about contaminants of emerging concern, and this relates to all of the chemicals that we use every day. You can see here, just in the last few years, we've added 85 million unique substances. This includes, some of the classes of chemicals that you see on the left-hand side here: anti-inflammatories, soaps and shampoos, pharmaceuticals. You can see the other ones on the right-hand side. This is a photo. I'm kind of weird. Whenever I go to CVS, Rite Aid, the grocery store, I look at all the products, I turn them around, I take photos of the active ingredients, just so I know what's in some of the substances that we use. Now, ultimately, those substances go down the drain, they get into the environment, from wastewater treatment plants, agriculture, other sources. And one of the big questions with these chemicals is, since they've been so carefully designed to have biological kind of considerations in our body, what do they do with the environment? One good example of an unintended consequence is diclofenac. This is an anti-inflammatory used to treat cattle. In India and Pakistan, when cattle died, the vultures would eat the carcass. And it turns out that diclofenac is actually super toxic to vultures. Populations decreased, by over 97% just in a few years. Another class of chemicals that we're concerned with are endocrine disrupting chemicals, especially estrogenic hormones, like 17-alpha-ethinyl-estradiol. This is the active ingredient in the birth control drug. And the plot here is basically showing when you introduce very low levels of birth control drugs into lakes, fish populations can crash. One more here is the UV filters or sunscreen chemicals. These have been of increasing concern in the last few years due to potential toxicity in corals. Some of these chemicals have actually been banned from being sold in certain states due to these concerns, which are really critical, for biodiversity, economics, as well as tourism and fisheries. And then finally, it took us 5 or 6 slides, but of course, PFAS is going to be present today. You're going to hear more from the other speakers too. We know there's thousands of different PFAS. They're in a lot of different products, and there's upcoming regulations that are going to be very challenging to meet. So all of these different contaminants of emerging concern, they can get into the environment from different sources. This is a figure from the EPA that was put together in the early 2000s that I really love. It was very forward thinking. They highlighted a lot of interesting sources here, some of which have been well studied, some of which have been a little bit understudied. And that's one thing that I want to try to emphasize as we move forward here. Maybe some of the more well-studied sources are wastewater treatment plants and agriculture. Some of the less well-studied sources I want to talk about are kind of present in the left-hand side of this figure. You see septic tanks, you see sewage lines, and these can end up leaking, and being important sources of emerging contaminants into the environment. Hey, Marissa, it looks like we're stuck on this slide.
Hold on one second.
Okay, so following from there, I would want to say one thing that I'm kind of excited about is, where I live. So I live in Baltimore. We're right on the Chesapeake Bay. This is really an interesting test bed for us. The western side of the bay, we have a lot of urban areas, suburban areas. Wastewater, septic systems are big sources. On the eastern side of the bay, we have a lot of animal agriculture, and this can also be sources of CECs like antibiotics and hormones into the bay. One thing that we've been interested in doing over the last 5 or 6 years is trying to understand those sources. Typically, we start with indicator chemical, sucralose. Maybe you put sucralose in your coffee. This is a synthetic compounds and artificial sweetener, and it serves as a great tracer for wastewater because it doesn't occur naturally. So we kind of looked at our sucralose concentrations around the bay. We were actually surprised. We thought wastewater treatment plants were going to be like kind of the major source correlating to sucralose concentrations. But what we actually found is when we sampled more diverse areas across the bay, septic system density was actually better correlated to sucralose levels. And septic systems aren't something that many environmental engineers think about all the time, but 20% of Americans are on septics. The data on this slide is showing that we found a lot of different CECs at high frequencies in septic wastewater and also at very high concentrations. So we're the weird people. We're going around knocking on doors, asking people, hey, can we sample your septic system? And thankfully, people were pretty open to that. Coming back out into the Chesapeake Bay here, here I'm plotting antibiotic concentrations. We were measuring 50 or 60 different antibiotics. So this is kind of the sum. Generally, they were present at low concentrations, except for a couple of areas where we started reaching like around 100 nanograms per liter. And this is where you can start selecting for antibiotic resistant bacteria. And so that's a huge public health concern, something that we're interested in trying to limit in Chesapeake Bay region. And then finally, I'll just mention, we weren't just sampling water, we were also sampling sediment and oysters from the bay. The UV filters are a little bit more hydrophobic. They partition well into oyster tissue. And so here's concentrations of one of the sunscreen chemicals that we're interested in. So the findings that septic systems were really important for CEC levels in the bay actually kind of motivated us to come back up, into urban areas. So here we're sampling in Baltimore in urban streams that don't receive wastewater effluent, but are around a lot of sewer infrastructure. And so we were worried that sewer overflows or leaks could introduce emerging contaminants into some of these streams. The columns that you see in the map here, those are all different CECs, kind of showing that we're seeing high concentrations and a diversity of chemicals in these urban streams, again, that don't receive wastewater effluent, but as you can see in the photo, the sewer infrastructure is right next to the streams. It basically runs along the streams. So any leaks or overflows can introduce these chemicals into the environment, pretty high concentrations. And actually, if we come back to septic, so this is a little bit further up in these watersheds, all of the dots in this map are septic systems. And we actually found that CEC levels correspond to septic system density. And so this was kind of critical for us because septic systems, in these more rural areas, thinking about wastewater leaks being important sources of CECs, these are ideas that haven't been explored so much. We usually kind of go to the more expected sources. I'll also just say in these urban streams, the CECs get into the water. We wanted to look at similar biomonitors, but we don't have oysters in these streams, so we use crayfish. And sure enough, we found that Baltimore crayfish are on birth control and they're using sunscreen. Finally, I just need to thank my group. They're all shown here. They're a great bunch. They work really hard. They're very positive and supportive of each other. And I get the pleasure of sharing out all of their results. So thank you very much and be happy to take any questions.
Lee, thanks so much. I love that. So you found crayfish that are on birth control and using sunscreen. Fantastic. I love that. Actually, there were some questions that came in, but I want to just kind of, hit you with kind of 3 questions so you can kind of get your mind around. We can talk through them a little bit. One is indicator compounds, right? So sucralose, fate and transport, similar to all the compounds, or are we going to get ourselves in this problem similar to using E. coli to tell us that we had feces, right? So indicators are good, but there's also some concerns with them. So if we can address that. Second, is there ever—
Can I take that one first so I don't forget?
No, and I'll come back to them, but just this gets your mind around them. So second, when are we ever going to be done with this problem? It's a little bit of Alice in Wonderland moving through the looking glass, right? Like there's always a new compound that we're worried about. So does this ever end, or is it? And then third is TSCA, right? So how does TSCA help us to not get these things in the environment in the first place so that maybe we answer the Alice in Wonderland question because we just do pollution prevention or good stuff up front and don't get stuff out there. So first question, indicators, sucralose. Are we getting ourselves in a similar problem like we are with E. coli for source tracking and that kind of stuff? What are your thoughts?
Yeah, I got it. Good question. So we like using sucralose, and a lot of people like using sucralose because it's fairly persistent. It doesn't degrade or break down very easily, and it also transports pretty well, so it doesn't kind of partition to sediment. And so if we're kind of looking at maybe like a worst case scenario, these chemicals are getting into the environment, they're not degrading, they're transporting downstream. Sucralose is a great indicator for that, but you're absolutely right. Other emerging contaminants have very different chemistries. That's one of the reasons why, we kind of measure a suite of around 100 compounds right now. Some of the UV filters and estrogenic hormones, they're much more hydrophobic. They prefer to partition, into sediment and tissue. And so one thing that we've been thinking about actually is using different components of dissolved organic matter as indicators for these different classes of emerging contaminants. But it's very critical to think about the actual chemistry, of each individual contaminant. Maybe that's kind of the heart of your question, because we can't treat them all the same. And so sucralose is not going to be a good indicator for some of the UV filters. And if we go in thinking that it is, we're going to have problems, trying to kind of back all of that out to understand what's happening.
Perfect. And then maybe just a quick one, and we'll just keep it really quick, right? But this whole idea, there's always an emerging compound, right? I mean, it's like there's going to be a new thing. We're going to research it, right? But how about the pollution prevention, the TSCA, the let's keep the stuff out? Thoughts on that?
Well, actually, I'd like to go to your second question, because I think it's a very good one. There are thousands, maybe even tens of thousands of emerging contaminants. One thing that we've done in our lab is try to prioritize the classes that we think are most relevant to kind of real toxicological outcomes. Antibiotics, this is a huge public health threat. This is probably the last century where antibiotics are going to work, because we're developing resistance so fast. And so what do we do, and how can we slow that down a little bit by doing better wastewater treatment, or maybe even reducing, antibiotic use? We overprescribe antibiotics right now. We misuse antibiotics. We can definitely cut that down without greatly affecting our ability to treat infections. The UV filters, this is one that kind of seems innocuous. Like, we put them on our body in pretty large concentrations. You're in Florida right now, probably you're wearing some sunscreen. But there is evidence that they're toxic to corals at very low concentrations, and corals are super critical for marine ecosystems. And so they're already facing a lot of stress from climate change. If we put chemical stressors on top of that, that can lead to kind of catastrophic consequences. So we've tried to put our emphasis on these classes that are either seeing real toxicological threats or real regulatory kind of things coming down the pipeline, like is the case for UV filters and PFAS right now. But can you define TSCA for me? I don't know what that is.
The Toxic Substance Control Act, right? So kind of giving the pollution prevention. And not that I don't want to follow up on that one, but I am also sensitive to time. So
got it.
You'll see some in the Q&A, and folks, I do encourage folks to follow up with Dr. Blaney directly. Lee, fantastic, great presentation. Thank you so much. We appreciate it. All right.
Thank you.
Thank you.